For more than 60 million years, Tyrannosaurus rex has been nothing more than stone and bone. Or so we believed, but what if that belief was wrong? What if something hidden inside T-Rex fossils was never supposed to survive at all? Scientists have recently discovered evidence inside these ancient bones that challenges everything we thought we knew about fossils, about time, and about the limits of biology itself. This discovery was so unexpected that many experts initially refused to believe it. Yet the evidence was real. It was verified, and it was peer-reviewed. Today, we are going to explore what scientists actually found inside T-Rex fossils, why this discovery was considered impossible, and how it may change paleontology forever. For decades, fossils were believed to be nothing more than mineral copies of ancient life. The idea was simple and widely accepted. Once an animal dies, its organic material decays, and over millions of years, minerals slowly replace the original tissue. By this logic, no original biological material should survive beyond a few 100,000 years. Certainly not for 68 million years. Tyrannosaurus rex lived during the late Cretaceous period, long before humans, before modern mammals, and even before grass became widespread on Earth. And yet, recent discoveries inside T-Rex fossils suggest that traces of original biological material may still exist. If this is true, it forces scientists to rethink fossilization, molecular decay, and the deep history of life on our planet. Traditionally, fossils are understood as mineralized remains. After burial, groundwater rich in minerals slowly seeps into bone, gradually replacing organic tissue with stone. Over millions of years, the original biological material disappears completely. This model is supported by chemistry, physics, and countless fossil examples studied over generations. According to known molecular decay rates, proteins such as collagen should break down entirely within a few million years, even under ideal conditions. DNA degrades even faster, usually disappearing within tens of thousands of years. Because of this, scientists believed that dinosaur fossils could never contain original soft tissue, proteins, or biological structures. The expectation was clear. Dinosaur fossils should be lifeless stone replicas, nothing more. In 2005, paleontologist Dr. Mary Schweitzer from North Carolina State University made a discovery that would shock the scientific community. While studying a Tyrannosaurus rex femur recovered from the Hell Creek Formation in Montana, her team used a weak acid to dissolve part of the fossil bone. The goal was simple: remove the mineral content so the internal structure could be examined. What emerged was completely unexpected. Instead of crumbling into dust, the bone revealed flexible, translucent structures that closely resembled blood vessels. These structures could stretch, bend, and even return to their original shape. This was something no one believed was possible in a dinosaur fossil tens of millions of years old. The presence of flexible structures inside a T-Rex fossil directly contradicted long-standing scientific assumptions. Organic tissue, according to everything scientists knew, should not survive for 68 million years. Proteins are unstable. They are vulnerable to heat, water, oxygen, and microbes. Based on known chemistry, collagen, the main protein found in bone, should degrade completely long before reaching even a small fraction of dinosaur age. Because of this, many scientists initially suspected contamination. Some suggested the structures were modern bacteria or biofilms that had entered cracks in the fossil. Others questioned whether the fossil itself was truly as old as claimed. Skepticism was intense, and the discovery was met with widespread criticism. To address these doubts, Schweitzer and her colleagues conducted extensive testing. They used advanced techniques such as mass spectrometry, immunohistochemistry, and protein analysis to study the material in detail. Antibodies designed to bind specifically to collagen reacted positively with the fossil samples. The molecular signatures matched those of vertebrate collagen, not bacterial material. Independent laboratories later reproduced similar results in other dinosaur fossils, including hadrosaurs. The research was published in peer-reviewed journals such as Science and Proceedings of the National Academy of Sciences. These results strongly suggested that the structures were indeed remnants of original dinosaur proteins. One of the most important discoveries involved the role of iron. T-Rex bones are rich in iron, likely released from hemoglobin when blood cells broke down after death. Experiments showed that iron can act as a natural preservative by stabilizing proteins and slowing microbial decay. In laboratory simulations, tissues treated with iron survived far longer than untreated samples. This process is similar to how formaldehyde preserves biological specimens. This finding provided a plausible explanation for how soft tissue structures could survive far longer than previously believed, though the idea remains controversial. Scientists did not find living cells, nor did they find intact DNA. Instead, they discovered remnants of collagen proteins, flexible vessel-like structures, and microscopic bone cells known as osteocytes. These structures retained their shape, elasticity, and molecular markers consistent with vertebrate tissue. Importantly, no claims were made about resurrecting dinosaurs or cloning them. This was not Jurassic Park. The discovery was far more subtle and far more scientifically significant. It suggested that under rare conditions, original biological molecules can persist for tens of millions of years. This discovery opens new possibilities in evolutionary biology. Protein sequences can reveal relationships between species more accurately than bone shape alone. Some analyses suggest molecular similarities between dinosaurs and modern birds, supporting evolutionary theory from a biochemical perspective. Understanding preservation mechanisms also helps scientists search for ancient biomolecules in older fossils, including those from early mammals and reptiles. It even informs astrobiology, as similar processes could apply to fossils on Mars or other planets. Despite growing evidence, debate continues. Some researchers argue that contamination is still possible, or that the molecules found are heavily altered remnants rather than true original proteins. Others question whether iron preservation alone can explain such extreme longevity. But this debate is not a weakness of science. It is a strength. Scientific knowledge advances through challenge, skepticism, and replication. Over time, as more fossils are tested and more laboratories confirm results, scientific consensus continues to evolve. This discovery does not mean dinosaurs can be cloned. DNA degrades far faster than proteins, and no verified dinosaur DNA has ever been recovered. The fragments found are incomplete and unsuitable for genetic reconstruction. There will be no dinosaur resurrection, no living T-Rex, and no Jurassic Park scenario. What we gain instead is something far more valuable, a deeper understanding of how life leaves traces across deep time. As technology continues to improve, scientists may uncover even more molecular remnants inside ancient fossils. Advanced imaging techniques, improved chemical analyses, and stricter contamination controls are expanding what is scientifically possible. fossils may no longer be viewed as simple stone impressions of the past. Instead, they may be understood as molecular time capsules, preserving fragments of ancient biology across unimaginable spans of time. Each new discovery pushes the boundary of what we believe can survive across millions of years, reshaping our understanding of Earth's biological history. The discovery inside T-Rex fossils was not magic. It was not science fiction, and it was not exaggeration. It was careful, methodical science, challenging long-held assumptions. What scientists found was considered impossible, not because it violated the laws of nature, but because our understanding of those laws was incomplete. The T-Rex, long believed to be silent forever, still has a molecular story to tell. And as science continues to listen, the distant past may prove to be far closer than we ever imagined.